Hello everyone. Today I'll be presenting our results using the Ionic system to extract DNA and RNA from FFPE tissues, as well as the protocol modifications we made to improve RNA extraction. I've been doing nucleic acid extractions for decades, and I know some samples are more challenging than others. FFPE tissues, for example. These are biopsies that have been fixed in formalin and then embedded in paraffin. FFPE is great for pathologists because it preserves cellular structure for techniques like immunohistochemistry. But for molecular biologists, not so great, because formalin causes cross-linking and deamination of nucleic acids. And while storing FFPE at room temperature is convenient, over time, it leads to more DNA and RNA damage and fragmentation. Now, when you use a colon or bead-based extraction method, the damage gets even worse. Why? Because nucleic acids binds to a solid surface, then get stripped off, and in between, they go through multiple washing steps. That means more fragmentation and some sample loss, which can be a problem for small tissues like needle biopsies. For about 20 years, we used colon kits for DNA or for RNA. DNA was usually good enough for Sanger sequencing and PCR, real-time or not, as long as we kept the design short with small amplicons. But there were limitations. RNA often wasn't long enough for gene fusion detection, and for next-generation sequencing, NGS, we didn't always have enough DNA or library prep will fail. And, of course, long-read sequencing was completely out of the question. About 4 years ago, I heard about the Ionic system. Back then, it was called Purification before Bionano bought it. The concept behind Ionic sounded really interesting, so we decided to give it a try on FFPE tissues. The protocol is simple. There is a first lysis step where the tissue is digested with proteinase K and paraffin is removed using mineral oil. Then there's a nucleus treatment called lysis 2 in the protocol. Well, I'll just call it nucleus treatment in this talk. You use RNAs if you want DNA and DNAs if you want RNA with different kits. After that, the lysate is loaded on the isotachophoresis chip along with the buffers, and about an hour later, you have pure DNA or RNA. So how does it work? What is isotachophoresis? It's the principle of separating charged molecules in solution based on their electrophoretic mobility. Once the sample and buffers are loaded on the chip, an electric field is applied. Nucleic acid, shown in green on the picture, will move and concentrate between the leading ions in blue and trailing ions in red. Impurities, shown in yellow with the DNA here, that were with the nucleic acid, will stay behind in the trailing ion zone. After about an hour, nucleic acid, still in green here, reach the elution well and are ready for collection. There's no surface binding, no washing, no dehydration step, no bias towards fragment size or GC content. DNA or RNA remains in solution in their native state. The result? Minimal sample loss and less fragmentation compared to colon or beads kits. When we first tested the Ionic system during a long demo, we got excellent results for DNA extraction. We tried both recent FFPE blocks and blocks that were 5 years old, and compared the yields with 2 colon kits. Ionic constantly produced higher yield of DNA. We were especially happy with the results from older blocks because that had always been a challenge before. Yield is one thing, but what about quality and fragment size? We usually assess DNA quality using quantitative PCR with 3 different amplicon sizes. Here are the results for 500 base pair. They suggest that DNA extracted with Ionic contains more fragments of at least 500 base pair compared to DNA from colon kits, because the amplification ratio is higher than the yield ratio. And what about longer fragment? Here's an example of PCR amplification of 900 base pair for an old FFPE block. Amplification is stronger and cleaner with Ionic, while DNA from the colon kit shows some degradation. But how long can we get? We recently asked a lab with a bioanalyzer apparatus to check for us. We were happily surprised to see that with recent FFPE blocks, we saw fragments as long as the 17,000 base pair marker with an average of around 8,000. For an older block, it was less, but we still got about 2,000 base pair, which is not that bad. We don't have data for our colon kits, but the other lab showed us one of their sample with an average fragment size of only 400 base pair. Does it work better for our downstream applications? Yes, it does. For NGS, we currently use an amplicon-based panel that doesn't require much DNA. But even so, we used to have 10 to 15% of samples that were rejected because of low DNA yield or library prep failure. With the higher yields of Ionic, the rejection rate is now very low. Also, based on the number of sequencing artifacts in older FFPE blocks, which drop from sometimes thousands to just hundreds, we can clearly see that DNA quality is better with Ionic. We also recently got better result for our new capture-based NGS panel with Ionic. Another application where we saw a big difference is a forensic kit we sometimes use to confirm sample identity in case of doubt. It amplifies short tandem repeats to create individual specific markers. On the right, you can see that with the colon kit, the peaks are so low that interpretation becomes ambiguous. With Ionic on the left, the peaks are much higher, making interpretation clear and reliable. During the Ionic system demo, we also tried RNA extraction. Our first attempt wasn't convincing. It worked, but the results were about the same as with our colon kit. We usually assess RNA quality by real-time amplification of 3 different lengths of the housekeeping gene PBGD. Successful amplification are shown in green. The percentage of amplification wasn't higher with Ionic for any of the 3 lengths. Since we usually extract RNA from only 2 or 3 samples per week, and the Ionic chip is for 8 samples at a time, we kept using our RNA colon kit until about a year ago when it was discontinued. We tried another colon kit, but it wasn't as good. So we then thought about switching to the Ionic system for RNA, which worked fine, even if it means wasting some wells on the chip. Then we recalled something from our training. For DNA and RNA, it's the same chip and probably the same buffers. The only difference is the nuclease added to lysis buffer 2. The lysis times and temperature, however, are very different, especially the times. For DNA, we start the lysis the day before because it takes about 9 hours, while for RNA, it's only 1 hour. The same applies to the Ionic FFPE complete kit, which uses 2 chips loaded on different days. That got us thinking. Maybe we could coordinate the lysis steps, start the RNA samples later, and load everything on the same chip? Then we had another idea. With our old RNA colon kit, we had already tested that overnight lysis work better with the 3 hours written, work better with overnight lysis than the 3 hours written in the protocol. So we wonder, would using Ionic DNA lysis time and temperature also work for RNA? If it did, it would make things so much easier. We tried a few things. For example, if and when to use the different nuclease treatments. In the end, the method that worked best was also the easiest. We use only one kit, the DNA one, and we buy DNAs. The first lysis step is the same for all samples. For nuclease treatment, we prepare a mix of lysis buffer 2 with RNAs for DNA samples and a mix with DNAs for RNA samples if we have any that day. And then we load everything on the chip. One single process for both types of extraction. It's much more labor efficient than our previous column kit or even the standard protocol for the Ionic FFPE kit. And the best part, it works really well. Based on duplicate amplification of 3 lengths of the housekeeping gene, RNA quality is much better with Ionic than with the column kit. For each length, the percentage of successful amplification was significantly higher with Ionic. We were even able to amplify the longest length in almost half of the sample, which indicate less RNA fragmentation. It's an important advantage for some downstream application, for example, gene fusion detection. These results are also better than what we got during the demo, the previous slide, when we used the RNA lysis protocol with the RNA kit. RNA yield was also about 3 times higher with Ionic. However, and I don't have time to show you the data, this study confirmed that yield alone isn't a reliable indicator of RNA quality because it doesn't correlate with fragment size. In conclusion, Ionic DNA kit enables simultaneous DNA and RNA purification from FFPE samples using one kit, one protocol, and even parallel processing on the same chip. For RNA in our hands, using the DNA lysis protocol improve yield and quality. For those of you who are using the Ionic RNA kit, it could also work well or even better with the DNA lysis protocol, but we haven't tested it. This method saves times, saves labor, and deliver high-quality DNA and RNA in sufficient quantities for our downstream application. And DNA sizes for Ionic are suitable for long-read sequencing technologies. And finally, I would like to thank the people who helped me with this project. Annick for her work on the RNA part, Dr. Labonte for providing the samples, the former Purigen team for lending us the instruments during a long demo and their help with the early work, and finally Bionano for their interest in our poster at AMP and their invitation at this symposium. Thank you for your attention.